Welcome to Well Explained. Students, I'm here with Biology Class 9th chapter, the fundamental unit of life, that is cell. But before starting the chapter, please subscribe my channel and press the bell icon so that you don't miss a class from me. Whenever you will get a class or I will upload any video, you will get notification. So let's start with the chapter. Now cell is the basic unit of life. We all know cell is the fundamental basic unit of life. That is, every living organism is made up of cell. Now in this chapter, you are going to study about prokaryotic and eukaryotic cells, multicellular organisms, cell membrane and cell wall, cell organelles such as chloroplast, mitochondria, vacuoles, endoplasmic reticulum, Golgi apparatus, nucleus, and about chromosomes, their basic structure and number. In this lecture, I am going to deal with the history of the discovery of cells, the main components which are present in the cell wall, and the transport of substances across the cell membrane. Now coming to the discovery of cell. Cell was discovered by Robert Hooke, an English scientist, in 1665. He saw cells in a thin slice of cork with his crude microscope. Crude means primitive microscope he has invented, and with the help of that he saw these cells in a thin slice of cork. He observed as honeycomb-like structure. That is why he named them as cellulae or cells, which means rooms. His discovery indicated for the first time that living organisms consisted of number of small structures or units. Now to know the history of cells, we have studied in 1665 Robert Hooke discovered the cell. In 1674, Antony van Leeuwenhoek studied living cells for the first time. The cell discovered by Robert Hooke were in the cork cambium, and the cork cells are dead cells. But Leeuwenhoek studied these cells in living organisms for the first time. In 1831, Robert Brown discovered nucleus in a cell. In 1838-39, two scientists, M.J. Schleiden and Theodor Schwann, they formulated the cell theory, and 1855, Rudolf Virchow stated omnis cellula e cellula. Omnis cellula e cellula means every cell arises from a pre-existing cell. This is also a statement of the cell theory. Now the cell theory. The cell theory states all living organisms are composed of cells. All living organism means whether they are plants or animals or bacteria or fungus, any living organism, they all are made up of cells, and the organisms may be unicellular, that means made up of one cell, or multicellular, that means made up of many cells. The cell is the basic unit of life. That means without cell, no life can be there. Cells arise from pre-existing cells. That is, they are not derived from spontaneous generation. Only the division occurs in the pre-existing cells and the new cells are formed. The modern version of the cell theory includes the ideas that energy flow occurs within the cells. Hereditary information is passed on from cell to cell, and all cells have the same basic chemical composition. This modern version of the cell theory you will study in the higher classes. Now, what are living organisms made up of? Just as a building is made up of bricks, the body of all living organism is made up of cells. जैसे एक बिल्डिंग में आप सोचें कि बिल्डिंग किससे मिलकर बनी होती है, ब्रिक्स से, ईंटों से मिलके एक बिल्डिंग बनी होती है. तो bricks are the building blocks of a building. In the same way, the cells are the building blocks of all living organisms. Basing on the cellular organization, some organisms are made up of single cells and they are known as unicellular organisms. Example are Chlamydomonas, amoeba, paramecium, trypanosoma, yeast. They all are unicellular organisms. And organisms which are made up of more than a cell, they are known as multicellular organisms. Irrespective of unicellular or multicellular organisms, the cell perform similar basic functions for their survival. Means unicellular organisms हैं तो भी जितने life processes हैं for survival of a life or a living organism, they all are performed by the single cell. And if it is a multicellular organism, then there is a division of labor, but the same life processes are performed by the group of cells. Now, gadgets to study cells. Gadgets to study cells means how these cells are studied. Cells are too small to be seen by the naked eye. अगर हम ऐसे ही देखना चाहें cells को तो we cannot see them. Therefore, they are studied with the help of microscopes. Microscopes are high-resolution instruments that are used for observing fine details of very small objects. Now, two common types of microscopes are light microscopes or compound microscope that range from 100 to 1500. This we use in our laboratories, whereas electron microscopes whose magnification ranges from 1 lakh to 5 lakhs. Okay, these are the two types of microscopes. Now, shape and sizes of the cells. The shape and size of cells vary, but all of these are ultimately determined by the specific functions. Some cells can change their shapes like amoeba, WBC, but plants and animals, they have almost fixed shapes. Shape का मतलब ये है कि depending on the place where the cells are present, they have different shapes and sizes. Okay. Smallest cell is PPLO, pleuropneumonia-like organism. It is also known as mycoplasma. And the largest cell is of an ostrich egg, which we can see with our eyes, like this only. But most of the cells we cannot see without microscope. Longest animal cell is the nerve cell. And a micrometer or micron is 1/1000th of a millimeter. Now, unicellular versus multicellular organisms. Unicellular organisms, first of all, we will study about unicellular organisms. They are made of single cell, and this single cell is able to perform all the life processes like obtaining food, that is nutrition, respiration, excretion, growth and reproduction. They cannot be divided into cells. They are also called as acellular organisms. For example, amoeba and paramecium, etc. Now, coming to multicellular organisms, they are made up of many number of cells. The cells have a similar basic structure and similar basic life activities. Different shapes and aggregated to form different tissues. Means different shapes of or the group of cells which are similar in shape and function, they aggregate, they come together to form different tissues. These tissues form organs, and the organs give rise to organ system that perform special function. Examples are plants, animals, and fungi. Now, cell is the basic unit of life. It performs all life function like intake of food materials, that is nutrition, excretion, metabolism, respiration, etc. etc. Now, in the cell, in the multicellular organisms, to be more precise, they are made up of many cells. The cells which are similar in function, they come together to form a group which is known as tissue. That is, division of labor is there. But within the cell also there is a division of labor. How? Cell usually possess a number of components called as cell organelles. And these cell organelles, they are responsible for different functions, such as mitochondria is responsible for the respiration, chloroplast is for photosynthesis, clearing waste materials is for lysosomes, for protein synthesis there are ribosomes, है ना? तो there is a division of labor within the cell also. Each cell organelle performs different functions such as clearing waste materials, protein synthesis and lipid synthesis. Now, what is a cell made up of? A cell is a tiny mass of protoplasm which is surrounded by a membrane and is capable of performing all the functions of life. A typical cell is formed of three parts. These three parts are present in all types of cells. There is a plasma membrane to create the separation of the outer environment, nucleus for its control, it is the control center of the cell, and cytoplasm possessing the metabolic machinery. Our cytoplasm consists of all the cell organelles. All the cell organelles, they are contained in the cytoplasm. Now, what are prokaryotes and eukaryotes? Prokaryotes are a group of organisms that lack a cell nucleus or any other membrane bound organelles. Means इसमें जो न्यूक्लियस होती है उसमें न्यूक्लियर मेम्ब्रेन नहीं होती है. मीन्स एक सेलुलर न्यूक्लियस इज एब्सेंट इन प्रोकैरियोट्स. All the nuclear material, it floats in the cytoplasm, and the other membrane bound organelles such as mitochondria, Golgi bodies, lysosomes, they all are absent in prokaryotes. They differ from the eukaryotes which have a cell nucleus. Prokaryotes are the single cell organisms such as bacteria and are roughly 1 micrometer in diameter. The nuclear material, that means the nucleus is without the nuclear membrane, therefore it is known as nucleoid. The nuclear material of a prokaryote is known as nucleoid. Whereas a eukaryote is an organism whose cells contain complex structures inside the membranes. The defining membrane-bound structure that sets eukaryotic cells apart from prokaryotic cells is the nucleus, or nuclear envelope is there within which the genetic material is carried. So got it? प्रोकैरियोट्स का जो जेनेटिक मटेरियल या न्यूक्लियर मटेरियल होता है वो न्यूक्लियर मेम्ब्रेन के अंदर नहीं होता है. न्यूक्लियर मेम्ब्रेन इज एब्सेंट. Membrane bound organelles are absent in prokaryotes. It is floating in the cytoplasm and it is also known as nucleoid. Whereas in a eukaryotic cell, the defined membrane bound structures are there within which the genetic material is carried. That means nuclear membrane is present, as well the membrane bound organelles are also present in eukaryotes. Now the difference between prokaryotes and eukaryotes. Prokaryotes are small in size, 1 to 10 micrometer, whereas eukaryotes are larger in size, that is 5 to 100 micrometer. Organized nucleus is absent in prokaryotes. whereas organized nucleus is present with nuclear envelope, nucleolus is present, and several DNA molecules are also present in eukaryotes, whereas in prokaryotes, a single DNA molecule is present suspended in the cytoplasm and nucleolus is absent. In prokaryotes, membrane-bound cell organelles are absent, whereas membrane-bound cell organelles are present in eukaryotes. In prokaryotes, vacuoles are absent, whereas in eukaryotes, vacuoles are present. In prokaryotes, mitosis is absent. It is the division of cell, whereas in eukaryotes, mitosis is present. Example of prokaryotes are bacteria, cyanobacteria, whereas examples of eukaryotes are plants, animals, and fungi. Now, this is a typical animal cell. You can see the different organelles embedded in the cytoplasm, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus is there, mitochondrion, nucleus, ribosomes, centrioles for cell division, and peroxisomes are present. Now, cytoplasm. The contents of a cell except for the nucleus is known as cytoplasm. सेल मेम्ब्रेन के अंदर और न्यूक्लियर मेम्ब्रेन के बाहर जितनी चीज कंटेंट होती है इन अ सेल दैट इज नोन एज साइटोप्लाज्म. It includes cytosol, organelles, vesicles, and the cytoskeleton. Cytoplasm has two parts, cytosol and cell organelles. Cytosol is the fluid part of the cytoplasm. It is homogeneous. Homogeneous means एक जैसा होता है ये, and crystalloidal solution liquid that contains substances, water, ions, enzymes, vitamins, amino acids, carbohydrates, lipids, proteins, etc. Now, cell organelles. They are the submicroscopic structure which are specialized to perform specific cellular functions. These are embedded in the cytoplasm. Cell organelles may be bounded by a membrane as in eukaryotes, but prokaryotes lack membrane-bound organelles. Virus lack cytoplasm as well as membrane. Now coming to cell wall. It is rigid, semi-elastic, semi-transparent and protective covering which is present outside the plasma membrane in plant cells, fungi and prokaryotes. Means cell wall जो है सिर्फ plant cells, fungi और prokaryotes में होती है. It is not present in animal cells and it is present outside the cell membrane, means it gives extra protection to the cells of plant, fungus and prokaryotes. It is made up of cellulose in plants, fungal cellulose and chitin in fungi. Extra deposition of lignin, suberin and cutin may be present during secondary thickening as the cell wall provides extra protection. So in the secondary thickening means when it gets mature, there may be extra deposition of lignin, suberin and cutin. These all are chemicals which are present. A cementing layer called middle lamella is present between the walls of two adjacent cells. दो adjacent cells are present होते हैं. You can see in the figure, in the picture, when two adjacent cells, तो जो दो cells की जो walls होती हैं उनके बीच में ये middle lamella is present. Cell wall possesses small pores through which adjacent cells remain connected through plasmodesmata connection. Okay. Now, what is the role of cell wall? What is the function of cell wall? It provides shape to the cells. Number one, it provides mechanical strength to plants. It protects against pathogens and mechanical injury. It helps in the growth of the cell wall. The growth of the cell wall determines the growth of the cell. Cell wall prevents bursting of cell on endosmosis. Endosmosis means when the liquid or the water enters from outside to inside. The cell swells. When the cell swells, it may burst. But because of the presence of this tough cell wall, this bursting is prevented. It has the ability to withstand a lot of variation in nature. Now coming to plasma membrane. In all the cells, the three basic components which are present are plasma membrane, cytoplasm and nucleus. So first of all, we are going to study about plasma membrane. The outermost delicate elastic membranous covering of the cell that separates its contents from the external environment is called as plasma membrane. Plasma membrane is selectively permeable. Selectively permeable means it allows entry of certain substances, restricting the entry of others. It is made up of a bilipid layer and proteins are integrated in out and inside. Means bilipid layer means double lipid की दो layers होती हैं और proteins होती हैं in out and inside. They are integrated on both the sides. Small carbohydrates are attached at place to outer surface of lipids and proteins, and its function is to give shape to the contents of the cell. It acts as a mechanical barrier. It is semi-permeable or selectively permeable. It helps in endocytosis, recognition centers, flow of information, osmosis, cell continuity, and it is modified to perform special functions like absorption in microvilli. Now, transport across the membrane. We know membranes act as a physical barrier between the organelles of a cell and its cytoplasm and its surrounding environment. If this membrane is impermeable, that means substances cannot pass through the membrane. If it is permeable, both solute and solvent can pass through means solute solvent दोनों ही अंदर आ सकते हैं. If it is semi-permeable, the membrane permeable means it permits to the solvent but prevents solute's passage. In solvent तो अंदर आ सकता है but solute नहीं आ सकता in semi-permeable. And in selectively permeable, these are four different terms. Membrane allows the passage of solvent and some selected solutes. I think it is clear to all of you. Now, active and passive transport. Substances may pass across the membrane without or with the expenditure of energy. If this transport is with the expenditure of energy, it is known as active transport, and if it is without the expenditure of energy, then it is known as passive transport. So in active transport, substances pass the membrane with consuming ATP because energy in the cell is in the form of ATP, that is adenosine triphosphate. And in passive transport, substances pass the membrane without consuming ATP. Now what is bulk transport? Large amounts of substances and food particles pass across the plasma membrane, so it is known as bulk transport. The process of bringing the bulk of materials from outside by the formation of endosomes is called as endocytosis. Means अगर बाहर से अंदर transportation हो रहा है, then it is known as endocytosis. Bulk expulsion of materials as in secretion and excretion is called as exocytosis. Means expulsion means when it is going out, expelled out. This is secretion हो रहा है या excretion हो रहा है. That means the substances are coming out. This is known as exocytosis. Now passive transport. It is of two types, diffusion and osmosis. Diffusion. It is the process of movement of substances, solid, liquid or gas, from the region of higher concentration to the region of lower concentration, till it spread uniformly in the given space. We have studied this diffusion in chemistry also. It is the movement of substances, and the substances may be solid, liquid or gases, from a region of higher concentration to lower concentration. Now, what is the importance of diffusion? It helps in spread of different substances throughout the cytoplasm of the cell. Osmosis is a type of diffusion where only solvent is allowed to diffuse. Diffusion helps in the exchange of respiratory gases between the cells and their environment. Transpiration is diffusion process, and flowers spread aroma through diffusion to attract insects and other animals for pollination. So these all are the examples of diffusion, and what is the importance of diffusion you can see. Now coming to osmosis. Osmosis is the diffusion of water or solvent, means liquids, across a semi-permeable membrane from a region of higher concentration to a region of lower concentration. In the diffusion also, the substance were moving from a region of higher concentration to lower concentration. In osmosis is the same, but the only difference is in diffusion there is no semi-permeable membrane, whereas in osmosis there is a semi-permeable membrane. Also, diffusion can take place in all the three states of matter, that is solid, liquid and gases, whereas osmosis takes place only in liquids. Plasma membrane functions as semi-permeable membrane. Now, difference between osmosis and diffusion. It can take place in solid, liquid and gases. For diffusion, osmosis takes place only in liquid medium. There is no involvement of semi-permeable membrane in diffusion, whereas osmosis requires semi-permeable membrane. In diffusion, diffusion takes place up till the concentration of the two medium is equal, means it equalizes the concentration of substances, whereas in osmosis it is not necessary that the concentration of solvent on the two sides of the membrane equalizes. Different substances diffuse independent of one another, means solid, liquid, gases, they diffuse independent, means they are not dependent on the other substances, whereas in osmosis only solvent undergoes diffusion. Diffusion is not much influenced by the presence of other substances, whereas osmosis is influenced by the concentration and type of solute particles. Now, types of osmotic solution. Osmotic solutions are those solutions which can cause osmosis. There are three types: hypotonic solution, isotonic solution and hypertonic solution. In hypotonic solution, the osmotic concentration is lower than another solution. In isotonic, the solution has an osmotic concentration similar to another solution. And in hypertonic, the osmotic concentration is higher than that of the another solution. In hypotonic solution, the osmotic concentration is lower. So, the movement will be from the other solution towards the hypotonic. That means the thing will swell. In isotonic, there is no net gain or loss because the concentration of the two solutions is same. Whereas in hypertonic solution, osmotic concentration of the solution is higher than that of another solution. That means this solvent will move from the hypertonic solution to the other solution. That means the body will shrink. Types of osmosis. There are two types of osmosis: endosmosis and exosmosis. Endosmosis is the osmotic entry of water into the cell or system, whereas exosmosis is the osmotic withdrawal of water from a cell or system. When we place the cell in higher concentration, the water comes out of the cell by a process called exosmosis. When we place the cell in lower concentration, the water enters into the cell by a process called endosmosis. Does cell wall and plasma membrane are similar in function? No. Cell wall occurs only in plant cells, whereas plasma membrane occurs in plant and animal cells both. It lies on the outside of the cell. Cell wall lies outside, means it is the outermost point of the plant cell, whereas plasma membrane lies on the outside of animal cells and inside in plant cell. Inside on plant cells, why? Because cell wall is present outside. Cell wall is permeable, that means it allows everything to come and go out of the cell, whereas plasma membrane is selectively permeable. Cell wall is formed of cellulose, hemicellulose and pectin, whereas plasma membrane is made up of lipid and proteins with small amount of carbohydrates. Cell wall provides protection and strength to the cell, whereas plasma membrane holds the cellular contents and control of passage materials. Now nucleus. Nucleus is also known as the cell brain or the control center of the cell. Nucleus is generally present in the center of the cell. In a typical or a generalized cell, it is at the center. But in plant cell, as there is a big vacuole present, this nucleus is shifted to periphery. The dense protoplasmic body that contains hereditary information controlling cell activities as well as for transfer to the next generation is known as nucleus. It lies in the median or central position. In mature plant cell, it lies towards periphery. In prokaryotes, nuclear membrane is absent and it is known as nucleoid. Cells like RBC, sieve elements, they lack nucleus. Cells without nucleus cannot survive for long time. One nucleus is present in most of the cells, but some cells may have more than one nucleus. That means cells can be uninucleate or multinucleate. Now chromosomes. Thread-like structures which are formed by condensation of chromatin during cell division are called as chromosomes. They are referred as hereditary vehicles because chromosomes are made up of DNA and proteins and they are responsible for the transfer of characters from the parents to their offspring. They are also known as hereditary vehicle, means एक parents से उनके offspring means उनके next generation पे जितने भी transfer of characteristics होते हैं, that is with the help of chromosomes. A chromosome consists of two similar threads called as chromatids. Two chromatids are attached at the center by a centromere and on either side of the centromere, chromatids are called arms. Now, what is the role of nucleus in a cell? Nucleus contains all the genetic information not only for the cell but also the whole organisms. It functions as control center of the cell as it controls cell metabolism and cell activities. Ribosomes are formed by the nucleolus part of nucleus and these ribosomes are further responsible for the protein synthesis. Nucleus brings about cell differentiation by which a cell attains a specific structure and function. Division of nucleus is essential for cell division and reshuffling of chromosomes and changes in genes produces variations. Now, this is whole for the part 1. In the next part, we are going to discuss about the cell organelles which are present in the cytoplasm. But before ending, don't forget to subscribe my channel and press the bell icon so that you get the notifications whenever a new video is uploaded. Thanks for watching. Have a nice day.